of this famous geneticist Motu Kimura, he derived interesting models of what happens to genes and chromosomes and all if there's absolutely no selection. This is called neutralism. And the attraction of neutralism was that it made for some very beautiful mathematics, I think, most of it. But it turned into a kind of belief system. So from 1960 to, in some cases, to the present, there's spread this idea that maybe selection isn't very important, maybe things are just random. So you have light hair, you have dark hair, etc., blue eyes, brown eyes, blood groups. People have been searching for the causes, but the neutralist perspective was they don't have any causes, they're just random accidents of history. So I had one of his students one time, this was in Japan, at dinner, tell me quite seriously that giraffes didn't have a long neck to get high leaves. Their neck just happened to get long for no reason, and that's why they ate leaves high in the tree. So this kind of spirit of kind of neutralist nihilism has been strong since the mid-1960s until the present day, especially in Japan. Selection and the whole idea of evolution having some meaning and consequences came back really in the about 1970 because of this man, William Hamilton, sort of brought out and made explicit that there's two kinds of evolution, at least, that you have to think about. One is adapting to the environment, resistance to tuberculosis, skin color, coping with sunlight, and all of that. But the other kind of evolution is gains, is coevolution between individuals or groups, that my own fitness depends on what I do for my kid, because he's got a lot of the same genes that I do. That whether I'm aggressive or a nice guy, the payoffs to those strategies depend on what kind of society I'm living in. So one has to consider a lot of different actors, and this really rejuvenated a lot of evolutionary thought. And then, finally, the kind of dessert, we're getting dessert now, is the Human Genome Project. And instead of every little sequence being precious, DNA, details of our DNA are, coming out right off the internet like floods from 100 fire hydrants. Nobody can keep up with the volume of data. It's absolutely unbelievable. I just have this here to show you what kind of detail about human diversity we can get now. Every one of those letters there, I-T, I-T, E-S, F, all of those are people. So this is taking, I don't know, maybe 300 or 400 people, and with a nice big computer, computing how different every pair of people is. So for every pair there, it's easy enough to compute their genetic difference over, in this case, 500,000 markers. And then what you can do is throw the data in a computer, throw them on a two-dimensional map like this, two-dimensional piece of paper, and then pick up a person, move him somewhere at random, and see if that makes the total amount of difference among who's in your sample smaller, a better fit to the data. Several ways you can do it. You can do it iteratively, that's called scaling, or there's a least squares solution called principal components. And I gather this is done with principal components, and you get the best possible map, the best possible two-dimensional map of differences among people. And what I want to point out is what you get out of this map of Europeans, you get Europe back. Those purple ESs are all Spaniards. And the PTs are Portuguese. And you can't tell Spaniards from Portuguese, but you can tell French from Belgians, from Swiss, from Germans. You tell Irishmen from Englishmen. This just regenerates a map of Europe. And there's enough information in the map that if you're Inspector Clouseau and your two suspects for robbing the convenience store were, one was a Greek and one was an Italian, you can take some DNA and with pretty good odds figure out which one's the guilty party. Take a little saliva and you can tell a Greek from an Italian, from an Austrian, from a Hungarian, from a Pole, from a Russian, that you can't tell them apart very well. Differentiate the different Scandinavian countries. You can take a random European and most of the time put him within a couple 100 kilometers of his birthplace. This is how much information and structure there is in human differences in Europe. So this is mostly a pretty picture, but to show you the resolution of these data that are pouring out faster than anyone can cope with them. But this neutralist idea that was so popular starting in the 60s and still is, people also liked it because it fit right in with a kind of popular ideology from the social sciences that everybody was the same. There weren't any interesting human differences, and we didn't have to think about evolution or human differences because there weren't any. This is Stephen Jay Gould, who was a famous writer about evolution. He says, that's why the speed of cultural change works at orders of magnitude greater than anything possible with natural selection, and why natural selection has almost become irrelevant in human evolution. See the neutralism? You know, we may be different, but it doesn't mean anything. No selection going on. There's been no biological change in humans in 40,000 or 50,000 years. Everything we call culture and civilization we built with the same body and brain. Well, Gould was a little bit of a strange duck, but here's Ernst Mayr, one of the most famous biologists of the last century. Something must have happened to weaken the selective pressure drastically. We cannot escape the conclusion that man's evolution toward madness suddenly came to a halt. And this was the sort of thing that, you know, biologists, a lot of them wouldn't agree with this, but this is the kind of thing that, you know, every science journalist knew to be true. And it's still with us. On the other hand, unfortunately, new data from the Human Genome Project and related projects has thrown a wrench in the works. If we look at human chromosomes, there is a lot of evidence of what are called selective sweeps of genes whose frequencies have increased very rapidly in a short time, with a little bit of algebra and mathematics and optimism. We can estimate a lot about these, and just on the basis of looking at the data, it seems that the rate of human evolution in the last 5,000 or 10,000 years is many times greater than the rate of evolution between chimpanzees and us. That the speed of change in humans has increased a lot. We see, and I'll talk about how we see in a moment, a lot of regions where one new gene, one new mutant has just increased a lot. But what's interesting is we don't see a lot of places in our genome where a new mutant has completely swept through and replaced the old versions. We see a lot of things that have grown to high frequency and then stopped at 40%, 70%, 60%. And that's what it means by mid-range frequencies. But hardly any places where there's been replacement of new genes. And this is an interesting and current puzzle that people argue about in human genetics. Why are these incomplete sweeps? Well, one idea is that they're new genes and they haven't gone to fixation yet. They haven't completely taken over. Because all of this rapid evolution started about 10,000 years ago when the numbers of humans ballooned after we started farming, after agriculture. So that's a hypothesis on the table. Another hypothesis is that some mutation has happened, it has high fitness, it's really good for you if you got one copy of it, and one copy of the normal or wild type or the ancestral form. But if it gets common enough in individuals, remember we're diploids, we're made from a blueprint, but we're not made from just one blueprint, we're made from two, one from mom and one from dad. So if the reasoning goes that if an individual ends up with two copies of this new sweeping gene, he's in trouble. So the more common this new one becomes, the lower the fitness, and this stops the sweep. Yes? Say again?
Use your example about racing in the South.
Oh, I will. I'm going to show you a slide. Yeah. Well, one well understood example of this is sickle cell anemia. If you're in an area where there's hyperendemic falciparum malaria, if you have one copy of that broken beta chain, you're a carrier and you do a lot better, about 20% better in terms of numbers of offspring in the malaria area. But if you've got two copies of it, you're dead. So it may be that there's a lot of this going on. It's called balanced polymorphism. And the third possibility is that there's a lot of stooge effect. Now the stooge effect is named after the three stooges. If you remember, three of them trying to get through the same doorway at the same time. And they all get stopped because they can't get through. And the stooge effect would be that, for example, the sickling mutation has a big advantage in Africa, protection against falciparum malaria, but it never got much further outside Africa because they've got hemoglobin E, and which does the same sort of thing, protects against malaria, and the advantage of hemoglobin S disappears when it runs into a competing gene. So this is something that people are arguing about a lot these days. Let me show you what I mean by a sweep and how we know there's a sweep. This is 11 chromosomes, and from different individuals, and this may be, say, 50,000, each one I'm showing 50,000 nucleotide positions. And everywhere there's a difference